Let us take a look at the three different types of cartilage. And the first one I have here is elastic. So again, if we go back to our guide, again at the top we're at chapter 5, cartilage and bone. And I scroll down and we really can pick either the epiglottis or the ear. And maybe I'll do both for elastic cartilage and then make a video for the others. And so we can see at a low magnification for the ear that on the surface you're going to actually see stratified squamous tissue because it's actually the entire section of the ear. And so on a lot of your slides I can see that stratified squamous. I can also see there's some hair follicles. You can see some of the glands. You might have some fat in here as well. And so again, over here on the side, it's giving you a matrix and the different cartilaginous cells. We also have a perichondrium that we're able to identify on either side of the cartilage section. And so even if I were to start at the periphery, just again to kind of get our bearings, as I always say, and if I scroll down here, all right, and you can see already it has that kind of patterning of stratified squamous. It's kind of flipped over, to what we probably usually think of, because we always have the skin and the surfaces, the epidermal surfaces at the top, but that doesn't really matter because it's going around like the front and the back side of your ear. And so if we were to pull this tissue down, you would actually see, if we go to the other side, you would see the same thing. All right, so there's actually their big chunk of cartilage is right there. Kind of keep on scrolling down. Lots of connective tissue and whatnot in there, and blood vessels. And then look, right there, stratified squamous again. So this would essentially be the keratinized layer of the skin. You can see the basal lamina that is here. This entire pink section is going to be our stratified squamous. And I can even, let's see, brighten this up a little bit. And this is like remnant of an actual hair follicle. All right. And so if you were to scroll around, you would actually see different layers of essentially the dermis. You might see some glands and that kind of thing in there, typical of what we would see in skin, right? So if we kind of zoom back out here, what exactly are we looking for in the ear? And we want to be able to see something that's pretty uniform and kind of as a line, or maybe there's like chunky bits of this cartilage. But again, like if you feel your ear and if you take this kind of like outer section of your ear, the cartilage is not sectioned. It's like a large actual uniform piece of cartilage. And so that's pretty much what we're going to see. So if I go out even to low magnification, you can see this nice long strip of cartilage here. And this is elastic cartilage. Your cartilage here is, very flexible, some more than others. And we can pretty much ignore a lot of this other tissue. I mean, we could get in there and look at muscle and fat and all of that good stuff. And that's there, but we really are here to zoom in and look at the cartilage. And elastic cartilage has a definite different appearance than when we look at, let's say, something like hyaline or fibrocartilage. There is a similarity, right? There is this kind of uniform appearance of the matrix that has more of a smooth background. I always say it has this watercolor wash to it, and it does. So in this area, basically from this point all the way down to here is your section of elastic cartilage. And you can see at the periphery that there is that kind of watercolor wash that exists. So let me actually zoom in a little bit more here. And you can see, right, it's very, very uniform. And we're staining on this slide specifically for elastin, right? And so elastic cartilage has a very high percentage of elastin fibers that help to support that matrix. The other types of cartilage, like hyaline and fibrocartilage, have a higher percentage of collagen fiber that makes it a little bit more durable. And so there is collagen here, but primarily we have elastin fibers. And you can see this scattering of, in pink, yours in lab is going to be more like almost like a black or a dark purple. You can see this kind of scattering of fibers like all over the slide. And again, because we're specifically staining for that. The cells here are very, very close together. They're not kind of spread out in a uniform pattern that you would see with hyaline. And fibrocartilage, they kind of are these kind of zones, or they're kind of clusters of cells. And they are, there can be some that are spread out, but they're usually kind of in clusters. And this is just kind of this massive array of cells close together. Okay, sorry, my computer kind of flipped out there for a second. And so we're talking about the fibers in the background and how there's a scattering of fibers that you see here, and that the cells are really kind of interdispersed like between all of the different fibers. And if we were to go in here and take a look at some of the cells themselves, and so if we go ahead and kind of click and dive in here a little bit further, you can see that, and as it takes a minute to basically refocus, you can see again this background is all this kind of pink material here is going to be all that elastin. In between, there's going to be a matrix that does fill in between all of these fibers, and again, this is all that non-living material, right? So matrix is the non-living substance. So this is going to be a combination of like the filler material and also of the elastin fibers. The cells themselves, and we can make a border around these guys. You can see that there's kind of a color textural difference that is here. The nuclei is pretty obvious to see. The cell would be filling up most of this kind of chamber area. All of these cells are going to have a lacunae around them. And so again, that lacunae is a space or kind of a chamber around the cell. And so we'll see this as a consistent factor between cartilage and also with bone, that the difference between a blast and a site is the fact that the blast is actively making the substance. It's actually actively making cartilage or a bony matrix. And so the material it's making is basically butted right up next to that cell. And so basically be like the chondroblast or the osteoblast, and then the material right there. There's no spacing that is there. There is no lacunae or that open chamber. And all of these are going to have an open chamber. So these are all adult cartilaginous cells. So these would basically be all chondrocytes. The same thing would be true for our bone cells. They would just be osteocytes. And so the cells that we see here, and as we go along, even if we were to zoom in here, that's like kind of a perfect one. You can see the nucleus. Here is the cell itself. And then there's a little bit of this white around it, and that would essentially be the lacunae, right? And so, you might come across, like here are two spaces, like there's a little bit of cytoplasm. You're like, oh, there's no nuclei. Well, don't worry about the fact that there's not a nuclei. It's probably on this side of the screen or through the screen. It's somewhere in there. We just don't get to see every single cell membrane and every single nuclei, right? There's different ways that we cut these tissues or things that we just don't get to see. The lacunae, again, is going to be around the outer surface of that cell. So even if we were to go to either side of this hunk of cartilage, we're going to start to run into the periphery. And even here, you can see that we have cartilaginous cells. We have essentially mesenchymal cells that start to differentiate here to make the surface of the cartilage, and also to essentially start making the membrane or the perichondrium. So even as we're all the way out here, these are still chondrocytes. You can see that there are still lacunae around them. This is more of a uniform appearance out here. This is a kind of like really what when you think of cartilage matrix on a slide, this is typically what we would see first. This is probably the easier view. And then we kind of start staining for all of the fiber. But that nice watercolor wash that we see, these again are all cells. This is what we refer to as the inner chondrogenic layer of the periosteum. And again, peri meaning on the outside, or perichondrium, sorry, the perichondrium. So it's on the outside, and chondro means cartilage. And so that membrane basically kind of starts here. And you have some mesenchymal cells that start to differentiate and kind of meet on the outer surface. We run into the outer fibrous layer as we kind of head out. If you're looking at this, even if we were to kind of flip over and do hyaline cartilage, and you were looking at the perichondrium and the trachea, which we will do, it's a much like thinner little layer, of fibrous tissue that kind of surrounds the outer hunk of cartilage that exists there. But for most of them, you can typically see the interchondrogenic and outer fibrous layer of the perichondrium. So you're kind of like going from this area outward, essentially, to kind of form that outer surface or that outer layer. I'm trying to think of anything else to bring up here. I think that's primarily most of it as kind of a generic view on elastic cartilage. Right along the border here is that perichondrium. This is kind of that newer cartilage, and then more adult cartilage that is here. And again, this is all elastic. I think that's about it. All right, thanks guys. I will also address the other two cartilage types and make videos for those too.